Everybody, and today we'll start with a pathology series. And the book I have recommended is the Harsh Mohan. It's, I think, 13th edition. Yes, and I have made notes out of it. So it's the second chapter because the first chapter was introductory only, introduction to pathology, and the second chapter is cellular injury, adaptations, and cellular aging. It's the part one, and we will cover this chapter in series of lectures. Okay, let's get started. First of all, the definition of cell injury is that a sequence of events occur when the stress or the cause increase the cell's capability to adapt. If the cause is so strong, or it is so stressful that the cell's capability to adapt is exceeded, so it results in the cell injury. Okay, the etiology or the causes of the cell injury, the most common cause is the hypoxia, which is the deficient supply of oxygen, and it is the result of ischemia. Ischemia is the deficient supply of the blood to an organ, and it results in hypoxia because if the blood is not supplying properly, so as the oxygen. The second cause is physical agents, for example, trauma or any other physical injury or road accidents, etc. Chemical agents like toxins, benzenes, carbon tetrachloride, microbial agents like viruses, bacteria, immunological agents. We can see here like our own immune system attacks to our cells or any other immunological agent. Aging, iatrogenic causes, the causes which are caused by the physicians or the doctors itself, and idiopathic of the unknown cause. These are the some etiology, just a brief overview of them. There are two types of cellular injury. First is the reversible cell injury, and second is the irreversible cell injury. Okay, as the name suggests, that the reversible means that the if the insult or the stress is removed from the cell, it can return to the normal shape, normal structure, and normal function. But opposite to that, irreversible injury, if even if the stress is removed, the cell cannot the cell cannot recover its normal structural and functional capabilities, right? There are some aspects of reversible cellular injury. There are four of them. Okay. First is the decreased generation of the cellular ATP. As you know that the ATP generation is from the two sources. First is the within the presence of oxygen, which is oxidative phosphorylation, and the second is the anaerobic pathway, the glycolytic pathway in which the glucose is converted into pyruvate, and if the if the mediates like intermediates for the Krebs cycle are available, the shifts to the Krebs cycle. But if oxygen is not available, it shifts to the lactic acid pathway. Okay, decreased generation of cellular ATP. Second is the lactic acidosis intracellular. Of course, when it shifts to the anaerobic glycolytic pathway, there is automatically accumulation of lactic acid inside the cell, and it lowers the pH. And if the pH is lowered, so nuclear chromatin clumping occurs here. The third one is the damage to the membrane pumps. There are a lot of membrane pumps in the membrane of the cell, the cellular membrane. Sodium potassium ATPases is the most common of them. It constantly pumps sodium out, 3 sodium out, and in return 2 potassium into the cell. But when it fails, it leads to the intracellular sodium accumulation. Okay, it was normally transporting sodium out. Okay, but when it fails, the sodium starts to accumulate inside, and as the sodium is solute, so it attracts the water, and water influx occurs. Also, the calcium pumps excessive influx of calcium, and the calcium accumulates in the mitochondria, and it will damage mitochondria. I will discuss here in the calcium influx of irreversible cellular injury. Second is the reduced protein synthesis. The hallmark for the reversible cellular injury is the swelling. Okay, the swelling is most important thing you need to remember. The membranes of the ER, the endoplasmic reticulum and Golgi swells up. When something swells, so the ribosomes on the surface of the rough endoplasmic reticulum gets detached from it and dispersed in the cytoplasm. So the protein synthesis will automatically be disrupted because ribosomes are responsible for the protein synthesis. Okay. The other changes include for the reversible cellular injuries, the myelin figures. Myelin figures are like circular figures which we can see under the microscope, and these are derived from membranes of the ER, membranes of lysosome, membranes of Golgi. The second one is loss of microvilli. It occurs because of the swelling of the plasma membrane. Okay, so the microvilli present on the surface of the plasma membrane gets swelled, and there will be loss of microvilli. For example, if normal membrane is like this, and if it swells up, it becomes like this. So it's automatically loss of microvilli. Then blabbing of membrane. Blabbing means formation of small blebs. Okay, this is the blabbing. Up to this point, if there is the stress removal or withdrawal, the cell can return to the normal state and normal function. But the second kind of injury is the irreversible cellular injury. As I have told that the calcium pumps fail and calcium influx occur and they collects in the mitochondria. And if calcium is collected in the mitochondria, it disables its functions and formation of flocculent densities. What are flocculent densities? These are the calcium salts, calcium salts, and these are the hallmark of early ischemic injury. Ischemic means that there is the interruption of the blood supply to a particular cell or particular organ. So, if there is a ischemic injury and it is the early stage, so the flocculent densities will formed in the mitochondria, and these are nothing but the calcium salts. Okay. The second is the activated phospholipases. Why the phospholipases gets activated? When the calcium influx occurs, it activates the phospholipases, and these are endogenous because these are present inside the cell and they degrade the membrane phospholipases. Phospholipases. Ases means that the ases suffix is used for the enzymes, and the cell membrane is formed of phospholipids. Okay, phospholipids, the integral proteins and cholesterol, a bit of them. So if the phospholipases get activated, it starts to degrade the membrane phospholipids. Okay. The third one is intracellular protease activation. Mainly here the enzymes gets activated due to the calcium influx. The second enzyme is the protease. Protease, as the name suggests, that it have a function for the destruction of proteins, and proteins are present inside the cytoskeleton. Cytoskeleton is the skeleton for the cell, as the human skeleton. So it integrates, it provides the force for the cell to stand, and it composed of microtubules, microfilaments, and intermediate filaments, and they contain proteins like tubulins, integrins, and desmin. These are degraded by proteases because it has been activated by the calcium. The fourth one is the endonuclease. Endonuclease activation. It starts to destruct the DNA of the cell. There are three forms the nucleus can be degraded, and these are like stages. First stage is the pycnosis. Pycnosis means the condensation of the nucleus of the cell. Okay. The second is the karyorrhexis. It is the fragmentation of the nucleus. The last stage is the karyolysis. Lysis means dissolution. Up to this point, even if the stress is removed, the cell cannot return to the normal state because the nuclear damage. Okay. The fifth one is the lysosomal hydrolytic enzymes. Lysosomes are vesicles or closed membrane structures, and they contain several enzymes inside them. They have hydrolytic functions, for example, many of them: amylases, lipases, cathepsin. Cathepsin is a protease. Okay, DNases, cathepsins, lipases, glycosidases, a lot of them. And if the lysosomal membrane is damaged, so they leak out of it and they causes the digestion of cellular components and it causes the cell death. Also, when these enzymes are liberated out of the cell, they leaked into the serum. Serum means the blood, okay? And they can be used as the clinical parameters. For example, in myocardial infarction, the enzymes are aspartate transaminase, cardiac troponins, lactate dehydrogenase, it's the most important, and isoenzyme of creatine kinase. If these enzymes are seen in the serum, we would suspect of what? A myocardial infarction. The second is the acute pancreatitis. We see lipase in the serum, and in case of the liver damage, we see alanine transaminase and aspartate transaminase. We see them in the serum, and we can suspect what kind of early disease has happened to the body. Okay, this one was for the irreversible cellular injury. Here's the table from the Harsh Mohan, and it is the schematic representation of reversible and irreversible cell injury. First, in the mitochondria, the swelling occurs and flocculent densities are formed. In irreversible, there are large densities are formed. Okay, the membranes causes in the reversible blends the swelling of the cell. Myelin figures can be seen, and in the irreversible as well as myelin figures can be seen. For the rough endoplasmic reticulum, the dispersed ribosome, protein reduction, the swelling, and in the irreversible it becomes lysed, it is dissoluted mainly. Lysosomes causes the autophagy of the cellular organelles. This one is the mitochondria, and it becomes swollen and ruptured in case of irreversible injury. Cytoskeleton, the main skeleton for the cell, it contains microtubules, microfilaments, etc. It gets aggregated or degraded, and in case of irreversible, it becomes disrupted. Nucleus causes the clumped chromatin. Why the clumping occurs? Because the lactic acid accumulation and the pH starts to reduce, and it causes the nuclear uh, clumped chromatin. In irreversible stage, pyknosis is the condensation, karyorrhexis is fragmentation of the nucleus, and karyolysis is the dissolution of the nucleus. Okay, I hope you understand the difference between reversible and irreversible cell injury. Okay, the depending on the duration of ischemia. Ischemia means the blood supply is disrupted, the reduction of the blood supply to an organ. Depending on duration of that, restoration of the blood results in three consequences. For example, if there is ischemia for a very short duration of time, and if there is restoration of the blood in this case, the cell restores the normal structural and functional state. Okay, it is not damaged in this condition if the ischemia is of the shorter duration. The second one is that if the ischemia is of a longer duration, the blood flow restoration is helpful. Neither it is helpful nor the reperfusion injury developed. What is the reperfusion injury? The reperfusion injury means that if there is the cell injury and if the blood supply is reperfused, the organ or the tissue or the cell is reperfused with the blood, it causes not stabilizing the cell, but contrastly, it causes further damage and causes the formation of reactive oxygen species. But in this condition, it is not occurred. The reperfusion injury not occurred in this condition, what we are talking about now, but it occurs in this condition. From ischemia to reperfusion injury, longer duration of ischemia, when the restoration of the blood occurs, rather than restoring, we would normalize that it would restore the normal structure, but it would not, and paradoxically, it causes the formation of reactive oxygen species. Okay, and these are the three consequences for the restoration of the blood flow depending on the duration of ischemia. Okay, the mechanism of the reperfusion injury has three aspects. The first, the most important, is the formation of free radicals. The second is the inflammatory reaction, and the third one is the calcium overload. Calcium overload means that if the restoration of the blood occurs and the calcium ions are also reperfused, and they causes the lipid peroxidation and membrane damage. These are the three aspects for the mechanism of the reperfusion injury. Okay, I will explain it to you for the free radicals, generation of free radicals. First of all, what are free radicals? Free radicals are the oxygen, mainly they are called ROS, reactive oxygen species, or any other radical that has charged ion. For example, the halide ions like nitrate, peroxynitrate, etc. These are free radicals. What is the mechanism for the generation of them? First step is the formation of superoxide. Superoxide, it is O2. Superoxide is formed by either by direct auto-oxidation of oxygen. Either by this step, the oxygen is converted into the superoxide by the enzyme known as xanthine oxidase, and cytochrome P450 inside the mitochondria. Okay, it is the first step for the formation of the superoxide, either by the auto-oxidation or by the enzyme xanthine oxidase and cytochrome P450 in the mitochondria, or it can be in the cytosol. The second step for the formation of free radicals is the formation of hydrogen peroxide H2O2. H2O2 is formed from the enzyme known as superoxide dismutase, and it is formed from the superoxide. The first step is the formation of superoxide either by autooxidation or by the enzymes. The second step is formation of hydrogen peroxide by the superoxide in the action of enzyme known as superoxide dismutase. Superoxide dismutase. What happens to the hydrogen peroxide? It is converted into the hydroxyl radical, and it is the most damaging ROS, reactive oxygen species. It is the most damaging one. It is formed by the Fenton reaction. What is Fenton reaction? The Fe3 positive is converted into Fe2 positive in the presence of oxygen, and the hydrogen peroxide is converted into the hydroxyl radical in this case. Or the hydrogen peroxide can be reduced into the water by the enzymes catalase and glutathione peroxidase. These enzymes, catalase and glutathione peroxidase, these are beneficial for the cell or not? Just think logically, because the species, which is the reactive species, is being converted into water, and water is good for us. So these enzymes, catalase and glutathione peroxidase, are beneficial for the cell. Okay, but it can also be converted into the hydroxyl radical, and the hydroxyl radical can also be formed by the radiolysis of the water. Okay, did you understand this mechanism? First is the formation of superoxide, second is the formation of hydrogen peroxide, The third is the formation of hydroxyl radical, and it is the most damaging of them. Okay, what is the cytotoxicity for the free radicals? The free radicals are unstable and because they contain the charge, and the rate of destruction is determined by the action of enzymes, superoxide dismutase, catalase, and glutathione peroxidase. These three enzymes are used. If these are not degraded, these are highly destructive to the cell, and this is called as the oxidative stress. Okay, what is mentioned in the text is that the severity for the action of these radicals depend on the action of these enzymes, because these enzymes are constantly causing the free radicals to be converted into the water. But if they are not functional, so they become more toxic to the cell. What are the toxicity for the free radicals? To the proteins, wait a second, to the proteins they causes the degradation of the cytosolic proteins and accumulation of denatured proteins in the cell. They causes the change in the amino acid residues, for example polymerization, fragmentation, conformational changes, and they produces abnormal proteins. For the lipids, they causes lipid peroxidation, membrane damage because the membrane is formed from the phospholipids, and TBARS. What are these? I will mention here. Wait a second. The third one for the DNA, they cause the altered gene expression and they cause single strand breakage for the DNA. Conclusion is what? That free radicals are very damaging to the cell and they causes all of these diseases. For example, cancer, they cause aging, neurodegenerative diseases, hypertension, inflammation, liver damage, etc. What are TBARS? It is known as thiobarbituric acid relative substances. These are formed as the byproduct of lipid peroxidation. Okay, and these are also very damaging for the cell. If these are present, if we see TBARS in the cell, we would think of the lipid peroxidation, and lipid peroxidation is caused by the ROS, reactive oxygen species. Antioxidant are the substances that inactivates the free radicals, and these are very beneficial to the cell because they causes the inactivation of ROS. Vitamins are A, E, and C, the ascorbic acid, vitamin A and E, sulfhydryl compounds like cysteine and glutathione. You have heard of glutathione, like many cosmetic fields use glutathione injections and tablets for the brightening of the skin, because the main aim is to get rid of free radicals. Serum proteins like ceruloplasmin and transferrin, these are antioxidants and these are beneficial for the cell. Other free radicals include nitric oxide and peroxynitrate. Nitric oxide is formed in the endothelial cells and it is a vasodilator in the endothelial cells, in the macrophages, and in the neurons. And with the help of superoxide dismutase, it can be converted into peroxynitrate. The other free radical is the halide reagents. These are released by the leukocytes. These are chlorine or chloride, bromine or bromide, and they can be combined with the superoxide and can form hypochlorous acid, HOCl, and it is also very damaging to the cell. Okay, up to this we have covered the reversible and irreversible injury. We have covered the mechanism of the reperfusion injury by the free radicals, mechanism for generation of free radicals, and you can see here that the effect of free radical is the protein oxidation, DNA damage, lipid peroxidation, and cytoskeletal damage. Okay, the next topic is the stress proteins. What are stress proteins? When the cell is exposed to any type of stress, the it will lead to the formation of stress proteins. Stress proteins are of two types. First is the heat shock proteins, and the second is the ubiquitin. What are heat shock proteins? These are the intracellular carrier proteins and present in the renal tubular epithelial cells, like in the kidney, and they normally work as molecular chaperones. What are chaperones? Chaperones are the molecules which assist the folding and unfolding of large proteins. Like these are involved in the protein management, like we can say that, and they causes the metabolic substances guidance to the site of their activity. They they are involved in the protein kinases, okay, and in response to the stress they are damaged, their level goes up and they leak into the plasma. Heat shock proteins, what are them? They are intracellular carrier proteins. They work as molecular chaperones. They assist the folding and unfolding of proteins. They guide the metabolic molecules to the site of activity, and they causes the protein kinases, the transport of proteins, okay. But in response to the stress or various insults, their level goes up and they leak into the plasma. The second stress protein is ubiquitin. Ubiquitin is a small protein about 76 amino acid, and it is very important because it determines the fate of many proteins in the cell. What is protein clearance? Protein clearance means that if the protein is of no usage, or if, for example, if it is dysformational protein, the ubiquitin causes the protein clearance from the cell. But in case of the ubiquitin dysfunction, the accumulation of protein inside the cell occurs, and it is the basis for the many neurodegenerative diseases like Alzheimer's, like Parkinson's, because it is a result due to the dysfunction of ubiquitin proteasome system. For example, phosphorylated tau, it is the hallmark for the Alzheimer's disease, and it is the evidence for the deficit of the ubiquitin protease system, and it causes the p tau accumulation. The conclusion for this paragraph is that the ubiquitin proteasome system causes the protein clearance from the cell, but if the system is dysfunction, it causes the protein accumulation, and it is damaging to the brain, causes the neurodegenerative diseases. Okay, the last is the like some brief explanation for the chemical injury. There are many chemicals like mercury chloride. It causes the poisoning for the alimentary canal and the kidney damage, causing the dysfunction for absorption and excretion respectively. Cyanide, it poisons the mitochondria, cytochrome oxidase, and this is the enzyme used for the oxidative phosphorylation, so it will not occur. Chemotherapeutic agents like for the cancers, heavy metals like lead, iron, these are the chemicals. The others includes the carbon tetrachloride, bromobenzene, acetaminophen, which is the antibiotic, but the high dose of them can cause destruction for the cell. The CCl4 is a very classic example. It is converted into the CCl3 by the body's P450 enzyme system in the liver cells, and it is very damaging to the cell. Okay, I hope you understand this. Uh, this is the ionizing radiations. They causes the radiolysis of the water to the hydroxyl radical, and hydroxyl radical is a ROS, reactive oxygen species, and causes lipid peroxidation, DNA damage, genetic damage, etc., cell membrane damage, all of them. The last thing is this diagram, and this like schematic diagram. First is the ischemia. This is for the injuries. Ischemia results in hypoxia. ATP is decreased. The production of ATP is reduced. Cytosolic calcium accumulated because the calcium pump are not working, and they accumulate in the mitochondria. They cause phospholipase, endonuclease, protease activation, and they cause the cytoskeletal injury, lipid breakdown, membrane damage. The reactive oxygen species like superoxide, hydrogen peroxide, and hydroxyl radicals, they cause the lipid peroxidation and DNA damage. The membrane phospholipid synthesis is reduced and it leads to membrane damage. This one is like the schematic representation for what is happening in case of ischemia and hypoxia. I hope you understand this topic, and this was very, very hectic and very long, but the main aim is to understand this. Just remember the some hallmarks for the reversible and irreversible injury, and the difference between them, the function, the cytotoxic damage in case of ROS, and the heat shock proteins and ubiquity. And thank you for today's lecture, and we will meet you in the next lecture. Until then, goodbye.